Hello, my name is Carla Gómez-Montes, and today I will talk about the first part of my PhD project, which is integrating ZooMS and Taphonomy to reassess the Paleolithic and human carnivore activities at the Mirón Cave. First of all, I would like to say that this is part of the ERC project Un Bailey de Sado, Illuminating Late Pleistocene Human-Carnivore Interaction in Europe. And as it says, it wants to reassess how these human and carnivore interactions were taking place. And in my case, my PhD will focus in identifying the unseen proteomic and taphonomic approaches to human-carnivore coexistence exactly in the Franco-Cantabrian region during the Upper Paleolithic. So I will try to assess this question in the Franco-Cantabrian region in different cases that I will present next to this slide. And we reached a co-tutelar agreement between the University of Vienna and the University of Cantabria, and Pere, Gelabert and Annabelle Marín-Arroyo will be my supervisors. So these are the three caves that will be part of my research, which is El Mirón and Jonim from Spain and Isturiz from France. Today I will focus in El Mirón, which is located, as I said, in the Iberian Peninsula, exactly in the Cantabrian region. And here we can say from Google Earth how it looks like and where it is located exactly. So El Mirón Cave was discovered in 1906, and after that there were taking place several small archaeological campaigns, but the proper ones started in 1996 with Straus and González Morales. Also, El Mirón, it is known by the Red Lady, which was founded inside of the cave. It was a woman that was buried there and was covered by a stone, and her bones were founded with ochre into it, and that's why it is called the Red Lady. My samples come from a part of the vestibule, which is called El Corral, exactly for two different squares, which is the U10 and the X10. And regarding the stratigraphy, from the X10 and the U10, I took samples from the levels Gravettian until the Magdalenian, which is from the 119 until 128. So all of this research will be my first paper out of the three main ones that I have to do for the University of Vienna. And it will compare three different data sets. The first of all will be the archaeofaunistical and taphonomical record, which has assessed by now that these are the species that has been found archaeologically in the settlement. And as we can see, only two carnivores have been found, which are the wolf and the fox. Also, the study made by Gilling et al. In 2025 shows how the taphonomical record look like, and the carnivore activity is quite limited, but still we would like to assess which were the animals or which were the carnivores that were making these affections to the bones. Then we would like to compare it with the ctDNA. By the moment, the ctDNA has been done by Gelabert et al. In 2025, but also study of my colleagues Larissa Barstch will continue investigating the site of El Mirón. They found the species that have already been found by the archaeological record, but also new species that haven't been found until now, which are these ones. But in my case, I will try to focus in the carnivores one, which are the hyena, the lynx, the quon, and the leopard. And we will try to find them applying ZooMS. So by the moment, I've taken 363 samples from El Mirón. Also, I've taken all the samples from the other two caves that will part of my PhD. And I've taken them from the unidentifiable bones of the records of these caves. And I would like to explain a little bit what is ZooMS. ZooMS is zooarchaeology by mass spectrometry, which is a technique in which one, after selecting the bones, which normally, as I said, comes from the unidentifiable bones, you apply this protocol in which you can extract the peptides of the collagen of the bone, and then you can introduce it into this machine, which is the MALDI, the mass spectrometry machine. and it gives you the result and that you can read with a program which is called MMAS, and they look like this. So each peak that you can see here is a different peptide. So each animal has different composition of peptides, and by comparing it to a database, you can know which animal have you obtained, or if you cannot arrive to the level of which exactly animal it is, you can arrive to the level of family. So we will try to find carnivores and human by this way. I've just finished the lab work for El Mirón in the Duca lab from the University of Vienna, where I've learned the whole protocol, and now I will start with the spectral reading. I will do it in two different ways. I will read it manually by using a MMAS program, and then I will learn to use a new way, a new approach, which was developed in 2024 by Berg and Duca, and it's called a species scan, which is like a systematic way of reading the spectra. So the main objectives and expected results of the research will be, first of all, identify these carnivores that have been present in the zDNA in the unidentifiable bones. And we expect to obtain these same carnivores in the zDNA that has been found in the zDNA, sorry. Also, we would like to determine which was the occupation that the carnivores were taking in the cave. It was sporadic, it was seasonal, maybe they were just passing through the cave. Also, we would like to attempt to find if there's human bones inside of the unidentifiable record, again by applying ZooMS. And finally, we would like to reassess which was the relation that the human and carnivores were having. They were sharing the space, it was seasonal, maybe it was only occupied by humans. As I said, the carnivores were just passing through the cave and not inhabiting in it. And I will try to solve all these questions soon. But in case that we don't obtain the carnivores bone that we are hoping to obtain, still this study will enlarge the faunistical and taphonomical record. It will be the first large-scale ZooMS study that had been done in the Franco-Cantabrian region, and it will enlarge the information about the human subsistence strategies and the animal adaptations in this period. Finally, I would like to acknowledge my supervisors and TAC members in the University of Vienna, all the collaborators of this project, and also the labs that I belong to right now, which is the EvoAdapta lab from the University of Cantabria and the Duca lab and BIM lab from the University of Vienna. And if you have any questions or comments, I will be happy to solve them, and you can contact me by email. Thank you very much for your attention.